received BRF funding in 2009. My first proposal that was funded was gene therapy for treatment of epilepsy. And this was a project that aimed to use ion channel genes to treat refractory epilepsy. So each of the two BRF grants resulted in federal funding for this research, for continuation of this research. So the 2009 grant, Gene Therapy for Treatment of Epilepsy, I received an NIH R21 grant. I received a second BRF seed grant in 2010 for another ion channel gene that's called TRPA8B, and that project was the role of TRPA8B in epilepsy. The second grant for the role of TRPA8B in epilepsy, I also received an R21 from the NIH, and the idea is that with these two-year grants, I would then be able to get enough preliminary data or ongoing data to justify a much larger award. The goals of the research that the BRF has funded have been to determine whether the feasibility of gene therapy as treatment for epilepsy. So that this was an important project whose ultimate goal would be to use gene therapy, so delivering genes to the brain of in the first stages epileptic animals, but later hopefully to use this technique to deliver genes to the brains of humans. And the second project was to identify a brand new target for a treatment of epilepsy. So basically to confirm the role of this protein TRPA8B in epilepsy, so that we could, as a second step, develop new treatments. The BRF led to very promising, tenfold amounts of funding for this important research.